I'm going to be talking about plant pathogens. So lots of people here are from the medical side, so when you hear about fungal pathogens, you might be thinking about Aspergillus or Candida. In agriculture, we might think, for example, of black sigatoka in bananas. We might think about Botrytis spray mold in soft fruits. And on cereals, we might think about Septoria, Ramularia, net blotch, and mildews. So here's some pictures of some of the diseases we deal with. It's a slightly prettier slide than some of the pictures medical mycologists like to share, but these are still incredibly damaging, both to crops and to human health too. So in a One Health context, plant pathogens matter because we're losing crops, we're losing crop yield and quality and shelf life, so there's more waste, less yield, poorer nutrition. They can also lead to toxin contamination of food, so mycotoxins such as aflatoxin or DON or ergot. There are also some concerns about the effects of the crop protection measures, so effects of fungicides and pesticides on humans and the environment, and some of the risks we've heard about with collateral selection of human pathogens in environmental niches. And also in terms of environmental health, if you're reducing the yield per area, then you might end up having to use more land, so there's a loss of natural habitats and amenities, as well as other resources such as fertilizers and water. But there is a positive side to the One Health approach. It means we can work together to share knowledge for shared problems. So in the case of fungal pathogens, that's the pathogen biology, epidemiology, management, and the reason we're here today, the antimicrobial resistance. And this is very much a universal problem. Agriculture, medicine, all fields, any effective control measure against a population is exerting selective pressure in favour of withstanding that control. And that selective pressure has some shared risk factors across agriculture and medicine. So if there's a lack of diversity of treatments, you're having repeated use of the same drug or the same mode of action. If you have either a susceptible patient or a susceptible crop variety, lack of diversity in the crops. So generally a lack of diversity in the host and in the control set against really very high diversity and adaptive potential in the pathogen that causes a risk of resistance. Now, when I say lack of chemical diversity, those who are on the medical side might be thinking, but wait a minute, we have a lack of diversity, you have all of these different groups. In practice, it's not such a positive situation. So, for example, if you're dealing with fungal pathogens, you can rule out to begin with any on here that are actually bactericides, any on here that are actually specifically controlling oomycetes. And for most pathogens, you can rule out any that just control mildews or just control one or two pathogens such as tacle or rice blast. So that's taking down the options. There are, as I've mentioned, concerns about some pesticides in the environment and for human health. So if we rule out any that have been banned or withdrawn or not approved in Britain and Europe, that narrows down our options. We take out any that are not approved so far. Some are closer than others. So we've got methyl-tetraprol, which we hope will be approved soon. We've got, for example, the DHO DHIs, which are approved in the US and Japan, but there are questions about dual use. Also, on the plant side, we don't just have many different pathogens, we have many different host species. So you'll find, for example, different sets of products are available for cereals or for broadleaf crops. Maybe some don't have good enough uptake in some crop species, or maybe some don't have the right spectrum of activity for the diseases on those crops. So, for example, if we're dealing with a cereal pathogen, we can take out any that are only for fruits, vegetables, and ornamentals. And suddenly the picture's not looking so rosy at all. So we have left, for example, the SDHI, so those are an important group in current use, but there is some resistance. We have the QOIs. They're still in use for some pathogens like rust, but a lot of others do have high levels of resistance now. We have the QIIs. They're a relatively new group. effective so far, but they are at high risk, so we need to be careful. We have the azoles, which we've already heard about in a clinical setting, but they're very important for plant pathogens too. We have morpholine, cyprosinil. They work for some pathogens, less active for others. We have multi-sites, which have protectant activity, but a lot have been lost due to environmental and toxicity concerns. Defense inducers, that's a growing area. The ones we have so far, they add some protectant activity, they're a useful addition, but won't do enough to fully replace the other fungicides. Biologicals, nothing on the market so far for a lot of cereal pathogens, but it is an area of active research. But as it stands, we do have limited options, and if you want to be mixing different modes of action and alternating different products between treatments, we certainly don't have options going spare, and we do need to find more options if possible. And the way we manage resistance, it looks a bit different from what might be done clinically, but sort of the same principles apply. So we want to be using the right product. We want to be mixing and alternating different products. We want the right dose so that we're controlling the disease, but not selecting for resistance more than necessary. We want to give the right timing. And we want to combine with any other available options, which for us will include things like resistant varieties, crop rotation and crop hygiene, and of course regular monitoring. So a lot of resistance management is about spreading the risk. So we want to use lower risk methods if we can, but also want to combine different fungicides, combine different varietal resistance. We want to be combining genetic and chemical and cultural control, and really diversify the whole system, so we're not having repeated monocultures of the same crop several years in a row. But when we're combining strategies, we want to do that based on the relative risk of each. So the lower risk strategies take more of the load, and then the higher risk ones are used just as needed. We have already said that actually any control measure will select for resistance, but that doesn't mean all measures are at equal risk. This will depend how complex an evolutionary step is needed from sensitive to resistant. It will depend how much variation is present in that pathogen population, and it will depend how strong is the selection for resistance. So the complexity of the step, that will largely vary between the control measures and the different target sites. Genetic variation will depend mainly on the pathogen, and then the strength of selection is the part we can affect through management. So if we have a really high risk fungicide, a really high risk pathogen, then we need to be stricter with the management, for example, limiting how many applications in a season and insisting that suitable mixtures are used. So when we're coming up with management guidelines for a product, for example, mixtures and alternations and dose rates, then we want that to be informed by knowing what is the risk level for that particular fungicide pathogen combination. Ideally, we also want to know about this earlier in the discovery pipeline to develop a lower risk target if we can, to know the risk ahead of release so we can be managing resistance from the start. And also, if we know what sort of resistance we're expecting, that can help with designing monitoring programs. So one of the main pathogens we work with is Zyme triticide, and this is a very diverse, very adaptable pathogen. So if we have the overwintering material, then we have ascospores, so these are sexual spores, so we have genetic recombination happening each year. And then during the growing season, we also have several clonal generations, so that's really increasing the population size and generating new point mutations in those clonal backgrounds, which will then recombine again in the autumn. And so with this very high adaptive potential, we have resistance to most of the single site fungicides. The most sites are low risk but have other concerns. MBC resistance was there by the 1980s. With the azoles, we have an ongoing shift, but we have had innovations within fungicide development, so we do still have some azoles that are effective now. the QOI's resistance occurred in the early 2000s. So in the UK, QOIs might be used on wheat for rust control, but they're not really contributing to control of septoria. SDHIs, again, we see some sensitivity shifts. Most isolates now would say are intermediate, but there are some highly resistant ones, especially with new very active SDHIs just coming onto the market. There's concern that those could spread too. And then the QIIs are the newest group, no resistance yet, but we are aware they are high risk, so we need to manage those carefully. And this is some work going through our isolate collection. So this is going back 20 years. So in this case, we have 5 different azoles, and you can see there has been a shift for all of them, but bigger shifts at different times for the different compounds. So chloride shifting from 2008 to 2013, tebuconazole, more shifts from 2013 to 18, epoxy and prothioconazole, really stepwise shifts throughout that period, small shifts individually, but adding up to a resistance factor of maybe 100-fold over that whole time. And the fenfluconazole, there is a shift from 2013 to 18, which is actually before this fungicide was introduced. But that's because there is some partial cross-resistance between different azoles. But these have different levels of intrinsic activity, so there have been shifts, but some of them will still give good control in the field. And here are a couple of examples of SDHI fungicides. So again, we can see gradual shifts over that time. Some differences in cross-resistance, but some shifts against both. I can go through these slides quite quickly now on resistance mechanisms, thanks to the excess introduction from Matt earlier. So again, a lot of these are the same that you would see in clinical fungal pathogens. So most commonly we would say mutations in the target site. So if that's SDH for the SDHIs, cytochrome B for QOIs, CYP51 for the azoles. We can also see target site overexpression, for example, due to CYP51 promoter inserts. We can also see efflux, so the fungicide is removed from the cell due to ABCR-MFS transporters. We see an example of that in septoria with the MG-MFS1 promoter inserts causing overexpression. So this is a non-target site mechanism. It can cause cross-resistance between different modes of action, but we also see a lot of isolates with multiple resistance to different fungicides due to combinations of multiple different target site mutations. And in terms of the predictability of resistance, when we're assessing that risk, trying to get ahead of the game, well, there is repeatability in the sense that resistance has emerged in multiple different pathogen species. We can also see at the genetic level, at the genotypic level, we see repeated evolution of the same mutations. So for the MBCs in beta-tubulin, it's E198 substitutions. Cytochrome B, G143A comes up a lot. And we see some recurring mutations for SDHIs and azoles, but we see a lot of rarer or one-off mutations as well. So here we can see the number of different mutations and how many species they occur in. So, for example, with the MBCs and the QOIs, we see a small number of mutations coming up repeatedly. So if we were to find QOI resistance in a new pathogen species tomorrow, more than likely it would be G143A again. Whereas the azoles and to an even greater extent the SDHIs, if you find another case of resistance, it could still be another new unknown mutation. And this really matters when we think about monitoring methods. And if we look at the azoles, this is just for septoria. These are just some of the different CYP51 genotypes that are out there. So starting with single or double point mutations. Earlier on, as we move towards the right, we can see up to 10 different point mutations in a single haplotype, in a single isolate. all leading to gradual accumulation of increasingly high EC50 values. So for my current fellowship project, I've been using experimental evolution methods to try and replicate resistance evolution in the lab and to assess that repeatability in different conditions. So I apply UV mutagenesis, I select with increasing doses of fungicide, and then take forward the populations from the highest fungicide dose where they're growing. And with this system, I'm asking, if you have replicate populations under the same selection, do they gain the same mutations? And then if you use different fungicides within the same class, is it all the same or different mutations? Does the starting genotype matter? Do the environmental conditions matter? And if we see more variable outcomes in the lab, does this correlate with resistance being less repeatable and less predictable in the field? So to start with, I looked at the MVCs. So these are the first single site fungicides. Resistance is really well established in the field. And so we can test our predictions against knowing what has already historically happened. So selecting with these 3 different fungicides, in most cases, most of the lines gained resistance. This was associated with mutations in beta-tubulin, so the target site encoding gene, a lot of different substitutions at codon 198 and at codon 200. And this does line up with what we see in the field. So in red, we can see all the mutations found in these lab lines. So they've all been found in previous mutagenesis studies in various different pathogens. They have mostly been found, or changes at those same codon positions have been found in some pathogen species in the field, although actually in Septoria in the field, we only see E198A, and that's not one that we've had in our lab population so far. So in terms of whether we can repeat resistance in vitro, yes, we've selected resistance. Yes, this is due to target site mutations, which is what we see in the field. Yes, a lot of those mutations are at the same codon positions in the field, but not necessarily the exact mutations. So in terms of what this can tell us, yes, it can show there's a risk of resistance. Yes, we could use that to test some general principles, for example, by alternating modes of action. Yes, we have the target site if we didn't know it already. Preemptive design of diagnostics. For a long time, that would have been a no. For example, we were using allele-specific qPCR. We need to know the exact mutation. Now we have more methods like amplicon sequencing, where it is enough to know the gene or the general region of the gene. But in terms of characterizing the exact mutations, so that, for example, at cross-resistance and fitness costs, that's the part we couldn't predict with certainty just yet. And this difference could either be because of differences in the mutations available in the lab versus the field, or differences in the selection pressure versus the field. So is it mutation limited, or is it because of fitness and differences in either selected conditions or the level of competition? So do we see, for example, that in the lab, we have a smaller population in the field, so we wouldn't necessarily have every mutation arising, but in the field, actually, there's more competition, so it's far more strict in filtering down to the fittest variant. And so I've tried to increase the mutational supply, not obtained E198A so far, but the next steps will be to look at different selective conditions. So I did also try some different azoles for selection. This is a case where it's actually been less repeatable in the field, so we'd expect it to be even less repeatable in the lab. As we've seen in the field, it's a far more complex situation. This is partly because we have incomplete cross-resistance, so mutations that are more resistant to one azole are less resistant to another. A historical example was between tebuconazole and prochloraz. We can also look at mefentrexate and prothioconazole, which are in the field now. So the least sensitive isolates to mefentrexate Some of them are also less sensitive to prothioconazole, but others, including the V136C change, actually are less sensitive. We also have epistatic interactions, so because it's multiple mutations accumulating, if you have a double or triple mutant, it could be either more sensitive or less sensitive than we'd expect based on the sum of its parts. So this means the genetic background matters and the selection history matters. So which azoles have already been used, which mutations have already been selected. So again, I use experimental evolution with a range of different compounds. And in this case, I've had a wider range of different mutations at different codons. So just so as we did find that A to C that we didn't get with the MBC, so it is possible in vitro. And in this case, we did have some that are the same mutations we found in the field, but actually with so many mutations in the field, it's a bigger target to aim for. We've also found some that are reported in other species and with other changes reported at the same codon. So yes, this is less repeatable overall than with MBCs, but still some of what we're seeing in the lab is recapitulating what's happened in the field. Slides in twice. So if we compare what's happened with the MBCs versus the azoles, we do find a wider range for the azoles, but I wouldn't say with either that without the benefit of hindsight, we could say at this point which of these many mutations in the lab would have been significant in the field. So in terms of the next steps, we'll be having a deeper look at what variants are evolving in the lab, looking over time, looking at perhaps rarer variants through amplicon sequencing. We'll be testing different selective conditions, temperatures, growth media, both with this experimental evolution with mutagenesis and also in competition assays with transformants with those different point mutations in a fixed background. We can also use whole genome sequencing to check if there are any non-target site mechanisms or any compensatory changes alongside those target site point mutations. And then once we have more of an idea in terms of matching what has happened historically, that's when we can be more confident using the system to predict what would happen for future control measures or to start getting into the what ifs, for example, of different azoles being used in different orders. So in terms of the implications for resistance monitoring, if we had known and highly predictable mutations, that's when we can get into these variant-specific diagnostics, so qPCR in the lab, or perhaps SNP-based LAMP assays that could be used out in the field. If we know the gene, but perhaps the exact mutations are more variable, more unpredictable, that's when amplicon sequencing comes into play, although we'd still want some functional and phenotypic testing alongside that to assess resistance levels for any new mutations or new combinations of mutations where there are epistatic effects. And then if we have completely unknown mechanisms, for example, if there's a non-target site or if we don't have a single known target site, that's when we are still relying on phenotypic testing, as well as the omic studies of the genomes, the transcriptomes, to identify mechanisms where we have variation in that phenotype. So yes, there is potential, and we have some projects now looking at high throughput testing, for example, with environmental spore samples and using next generation sequencing, but that does have to be alongside phenotypic testing as well. So I'd just like to thank all my colleagues, past and present, and the sponsors of the work in our group, and thank you for listening.